On this slide, we have a very nice graphic of what a neuron looks like. And you can see the little dendrites which receive messages from other neurons, the cell body which contains the identity of the cell basically, the mitochondria, endoplasmic reticulum, the nucleus, the ribosomes, the Golgi apparatus. And then coming down from that is a long axon. At the beginning of the axon hillock, you have myelin sheaths that go down in little sections all the way down the axon. And then you have the axon terminals at the end. If you look at the myelin sheath, it's interesting material. It's almost like the insulation you find on electric wires. You know, if you had a bare wire going across the floor, a lot of the electrical charge would dissipate into the floor. But because you've got the rubber surface, the rubber coating on the wire, electricity is prevented from diffusing into the floor and other things that it touches. The myelin sheath sort of works the same way. It prevents the charge from going into whatever's next to it. And they think it may be related to the complexity of the brain. They find that between the ages of 2 years old and 9 or so years old, there's a lot of development of myelin in children's brains, which may be why they're able to increasingly conceptualize and learn more and more complex ways of communicating in terms of language. The other thing that's kind of neat is that rather than having to run down the entire wire, which would take some time, though not much relatively speaking, the charge on these neurons is allowed to jump from node to node on the myelin sheath, which allows it to propagate the message much more quickly. Myelin can deteriorate, and in certain diseases like multiple sclerosis, it hardens and becomes plaque-like and doesn't protect as well. And that may be why the messages out to the muscles and back from the arms and legs don't get out as well in cases of multiple sclerosis. But I'm getting a little ahead of myself there. Moving right along, I want to talk a little bit about the neuron's structure. The cell body, as you saw, houses the nucleus, and it is basically the neuron's metabolic center. It provides the energy for the cell. The cell membrane is just the skin of the cell. The axons are that long cable extending from the cell body down to the dendrites. And what happens is the impulse from the cell body travels along the axon to its end, where the terminal buttons release neurotransmitters, which are chemical messengers, out into the synapse, and then they are received by other neurons after they pass the synaptic gap. And you're going to see an illustration of that later. The axon is, again, covered by myelin sheath, which is a fatty substance that helps impulses travel the length of the axon, as I described before. The dendrites are those little branches on the left top part of the picture that you saw before that extend from the cell membrane, and they receive neurotransmitters and messages via neurotransmitter from other neurons. So the path is that the neuron before sends messages that contact the dendrites, which propagate messages down the axon to the axon terminals, which release neurotransmitters that go across the gap to the next neuron's dendrites, and therefore convey the electrical message down the line, electrical and chemical message down the line. But more on dendrites. The adaptiveness of all the little branches is that they increase the receptive site of the neuron, and all sorts of contacts can occur along the surfaces of those dendrites. And here's another picture of an axon. You can see here the nodes of Ranvier, and these are the points between the myelin sheath sections, and messages can be propagated by jumping from node to node. You can see the axon hillock, the part of the axon that links to the cell body. And again, the myelin sheath is related, the deterioration of that's been related to multiple sclerosis. Now, in terms of what actually happens within the neuron, when a neuron receives impulses from other neurons, the cell membrane allows an open exchange of positively and negatively charged ions briefly. Action potential, which is a change in electrical charge, runs down the axon to the terminal buttons. And all this starts with the axon hillock, the gatekeeper of the neuron. The terminal buttons release the neurotransmitters, and then the neurotransmitters cross the synaptic cleft, the gap between the neurons, to the dendrites of the receiving neuron. Now I'm going to give you a little description of the process, and then I'm going to show you a little video clip. And I think this is probably what I find to be the trickiest part of the chapter. So it is worth repeating. Now the axon hillock is the gatekeeper. It determines who is the correct size for this sort of message. When the hillock receives a go signal, then it will release the potential into the axon, and then the charge will travel down the neuron and at the axon and across the cleft. And this is a one-way street. The action potential can't decide halfway through that it's going to change direction. It's not going back because there's a refractory period, and the axon hillock prevents it from going back. So the next node's activated, then the next, and the next, all in one direction. Put another way, during the resting state of the cell membrane, potassium and chloride ions are allowed to enter the cell, and sodium ions are not. The cell becomes polarized with the negatively charged ions inside and positively charged particle ones outside. And this electrical balance is called the resting potential. If a stimulus affects the cell strongly enough, the membrane temporarily becomes permeable to sodium ions, and the polarity changes. For an instant, the cell interior is positive and the exterior is negative. And this abrupt change is called an action potential and is conducted down the axon like a spark down a fuse, as you'll see in the clip. And then the neurons, after that whole process, restore themselves. Now these things travel extremely quickly, between 100 and 400 meters per second, or 200 to 900 miles an hour, depending on the properties of the neuron. And here's another picture of a neuron. Here are a few more characteristics of neurotransmission. Another thing is that it's all or nothing. The action potential either happens or it doesn't. It has to reach a certain intensity or it doesn't go, and once it goes, it goes. It's also non-decremental. Action potentials don't change in amount or voltage as they travel. There's just one unit that's acceptable that goes through. And there's a refractory period after, which I mentioned. Neurons need about 2 milliseconds to recover and rest before they can transmit again. There's also a threshold. The hillock determines what's a big enough amount for the message to be sent, and the minimum level of stimulation required is what's called a threshold. Once you reach the threshold, the action potential doesn't get bigger, it's just sent.
Now, I have a few video links that are in this module that illustrate things that happen at the synapse. And they also illustrate how synapses work. Some of the things that happen at synapses are many of the effects of drugs. And there are some different illustrations of how drugs are active at the synapse that are in your module. And there's also a couple about poisons, and sometimes the two overlap. So take a look in the module. Following this lecture, there should be some video clips there that will illustrate the specific interactions that happen that are caused by drugs at the synapse.
I'm going to talk a little bit more about communication between neurons. When the action potentials reach the end of the axon, they stimulate muscle fibers, glandular cells, and other neurons, and they have to transmit the impulse across a gap called a synapse. This is done by the release of neurotransmitters. These are stored in vesicles at the end of the terminals or endings of each axon terminals. When action potentials reach an axon terminal, they stimulate the release of these neurotransmitters, which diffuse across the synapse and activate receptor cells on adjacent neurons. And most nerve cells have thousands of synapses. Neurotransmitters are usually received by a dendrite, but sometimes they're received by cell bodies or axons. And there are two types of messages that can be carried. One is excitatory, which causes a reaction in the receiving cell or an impulse, and the other is inhibitory, which decreases or prevents a response or impulse. There are several different types of neurotransmitters. I want to mention a few notable ones here. One is acetylcholine, and this neurotransmitter affects the body's motion, memory, and attention, and may be involved in Alzheimer's disease. There's also dopamine, which affects voluntary movement, attention, and learning. High levels are associated with hallucinations, not just schizophrenia, but a lot of the drugs that cause hallucinations or paranoia increase dopamine levels. And on the other end of the continuum, too little dopamine levels has been implicated in Parkinson's disease. So if you have too much dopamine, you hallucinate. If you have too little dopamine, the messages have trouble making it to the muscles, and therefore you have more spastic movements. Endorphins are the body's natural morphines. They reduce sensitivity to pain and are linked to pleasure. They're sort of opiate-like. When you hear about a runner's high, what that is supposedly caused by is when you start running, your body starts automatic precautions to prevent pain. And once you hit the wall that they talk about, where the running gets painful for a while, and then suddenly there's this point at which it's actually pleasurable, that's when the endorphins have been released, and they're reducing pain and giving you kind of a runner's high. But they're a type of neurotransmitter. There's also serotonin, which is associated with arousal and sleep. And if your levels of serotonin are too low, you can suffer from depression. And Prozac increases levels of serotonin, as do a lot of antidepressants. In fact, SSRIs that you may hear about are selective serotonin reuptake inhibitors that keep a lot of serotonin in the synapse where it keeps you feeling good, instead of having it be recycled by the next neuron in the process of sending the signal. Norepinephrine is also associated with depression. If you have too low levels of serotonin and or norepinephrine, you're going to be more likely to be depressed. And norepinephrine is sort of the energy part. It's related to, used to be called noradrenaline. It's an adrenaline-like substance. So you're going to be really slowed down if you don't have enough of this. It helps control alertness and arousal, and again, low levels can depress mood. The next slide has a video that depicts neurotransmission, and if that doesn't work, there's also a link to it on one of the things in your learning, on one of the sheets in your learning module. Delete that. The next slide is attached to a video having to do with neural signal transmission. And if the video doesn't work, it's also part of your learning module number 2. Speaking of neurotransmitters, I thought I might tell you a little bit about how poisons work. Curare, which is a South American arrow poison, will get in and occupy receptor sites that the ACH or acetylcholine molecules normally use, and therefore prevent it from working. And so people will become paralyzed. With botulin, I've got a little video clip on that, but the way it works is that it blocks the release of ACH from the axon vesicles, and in that way results in paralysis. And finally, black widow spider venom will flood the neuromuscular synapses with ACH, which will do the opposite. There'll be too much activity, and there will be violent muscle contractions as a result. Of course, neurotransmitters can also be used therapeutically by, for instance, if you increase the amount of serotonin in the synapse, people's mood improves, and if you reduce dopamine levels, people's hallucinations go away. So a lot happens at that synapse, which is where these neurotransmitters act.